Hello, everyone. Can anyone hear me well?
Yes, very clearly.
Thank you. Very good evening to everyone, and welcome to INBIOSIS MSBMB webinar lecture series 1. I am Chyan, research fellow in the Institute of Systems Biology, and also a secretary for the Malaysian Society for Biochemistry and Molecular Biology. So I will be the moderator for today's webinar. So I hope just now some nice music and the video play to introduce University Kebangsaan Malaysia, UKM, and INBIOSIS. And so INBIOSIS started organized webinar since last year, as we know all the COVID-19. And for this special INBIOSIS MSBMB webinar, we will focus on special topics in structural biology and bioinformatics. And first of all, before we start, thank you for joining this first INBIOSIS MSBMB webinar. A webinar that a collaboration with Malaysian Society for Biochemistry and Molecular Biology between INBIOSIS and MSBMB. So before we start the webinar, please join me to welcome the director of INBIOSIS, Associate Professor Dr. Jeti Azura Muhammad Hussein, and President of MSBMB, Professor Dr. Leong Moy, who are also with us here today. And I would like to first invite Associate Professor Dr. Jeti Azura to give a welcome address, and followed by Prof. Dr. Leong Moy.
Thank you, Dr. Leong. Okay, can you hear me loud and clear?
Yes.
Okay, good. Right, so I would like to wish a very good afternoon and welcome to INBIOSIS MSBMB webinar to all our participants, to the distinguished speaker, Dr. Vinothkumar Kutti Ragunath from Tata Institute, Bangalore, and to the Honorable Professor Dr. Leong Moy, the President of MSBMB. So, and thank you so much to all participants. So as I can see now, we have about 123 participants on this Friday afternoon, and this is a very good turnout. I am very happy with your participants, and I would like to take this opportunity to thank all of you for the time spent with us, and I hope this 1 hour session will give you and will give all of us a new and interesting knowledge. Okay, so I, on behalf of INBIOSIS, are very honored to co-organize this webinar with the Malaysian Society for Biochemistry and Molecular Biology, or it is famously known as MSBMB. MSBMB, for your information, is one of the long-standing and prestigious local scientific community. So we are greatly honored with this joint venture, and we hope there will be many more to come. And I also hope that we will have a long-term partnership in providing a platform for interesting and new scientific knowledge to feed the hungry minds of our local scientists by inviting renowned and budding scientists from all over the world via our members network. I also hope that this partnership will be able to disseminate the knowledge, increase our motivation, and also enhance the scientific interaction between INBIOSIS, MSBMB, as well as scientific community, either in our country, in locally, or it can be expanded internationally. So this is our so-called survival mechanism that we need to be able to cope with, and then to cope with the increasingly challenging time during this pandemic. So I won't take much of your time because I know that all of us are very eager to listen to what Dr. Ragunath would like to share with us today. So again, I would like to take this opportunity to thank all of you for joining us. And then I would like also to invite you again to our many more webinar sessions that we are organizing. Okay, and thank you very much, Dr. Leong and the team, webinar team, and also the promotion team for your great effort in materializing our effort. Thank you very much. So everybody, hope you will enjoy and able to grasp and absorb new knowledge to be probably adapted in your scientific research. Thank you.
Thank you, Dr. Chetty, for the speech. I'd like to invite Prof. Lim. Yes, Prof. Lim.
Right, thanks, Chan Leong and Prof. Chetty Azura, the director from INBIOSIS, Dr. Vinothkumar, our first distinguished speaker for this webinar series, Dr. Ng Chan Leong, the moderator, fellow colleagues, organizing committee, as well as the participants. Very good afternoon and also evening to all of you. Right, so first and foremost, I agree with what Prof. Chetty has just mentioned. This is a very good platform for the collaboration between INBIOSIS and also MSBMB. We from MSBMB are very happy to have these collaborations between these two organizations. To have this INBIOSIS and also MSBMB webinar series. So when we hear the word series, meaning that we have many more exciting topics to come and cater specifically to our participants that are interested in this field of study. So we invite you for our future events, will be announced by Dr. Ng Chan Leong when come to the times. And thank you so much for joining this today's seminar. Right, and also for Dr. Vinothkumar, thank you so much for accepting our invitations to be our first, as I mentioned, speaker for this webinar series. And of course, we are actually anticipating to your very exciting sharing later on. I hope that through this kind of sharing and through this kind of platform, it will give us more opportunity for collaborations, not within the national institutions, but also international collaborations, like from your side, Dr. Vinothkumar. So looking forward to all these opportunities. Though this pandemic give us a lot of difficulties and limitations, but I think through the limitations, we create more opportunity, So we should stay in this kind of high spirits and looking for more opportunity rather than always think of the limitations. I think we will work very well for the past 1 year. I think we produced a lot of papers as well during this pandemic, though research actually being delayed for a while. So I think we were looking for more better conditions in the next few months with the vaccinations, which is ongoing process for all our nations in the current moments. All right, so to Dr. Ng Chan Leong again as a moderator from both sides, from INBIOSIS and also MSBMB, your hard work we are recognized, and also the teams that you leading, with the supervision from Prof. Chetty, to organize this webinar series. A lot of hard work have to put in, although we see it's only about 1 hour kind of sharing, but a lot of preparation work behind. So thank you so much, Dr. Ng Chan Leong, Prof. Chetty, for your leaderships and also lead the team to get this done very well. And last but not least, to all the participants, thank you so much for your continuous supports and participating in our webinar series. So I wish everybody stay safe and healthy and enjoy a very successful events today. Thank you so much. Back to you, Dr. Ng.
Thank you, Prof. Lim and both Prof. Chetty for the very warm welcome speech. So now I will give some housekeeping rules for regarding our webinar today. So the lecture will take 45 minutes and the Q&A section will take approximately 15 minutes. If you have more, I think Dr. Vinoth may willing to extend, right? And probably, so it depends on the number of our questions posed. So please send your questions or type your questions in the Zoom chat box or in the FB live page. So I will read the questions during the Q&A sections. So also I would like to remind before the seminar ends, I will invite all of you to switch on your camera for photography sections. So please stay tuned until the end of the sections to fill the attendance form for the e-certificate and also for as well UKM credit. Without further ado, now is the moment I would like to introduce our honorable speakers for today. Dr. Vinothkumar Kutti Ragunath from National Center for Biological Sciences, Tata Institute of Fundamental Research, Bangalore, India. Or more, Dr. Vinoth maybe like to call Vinoth, right?
Yes. Yeah.
Right. Dr. Vinoth has a long-standing background in membrane protein biochemistry and biophysics, in particularly using cryo-electron microscopy or cryo-EM to study structure and function of protein or membrane protein. Let us welcome Dr. Vinoth to share his experience and findings from his research. And Vinoth, Dr. Vinoth, the Zoom is yours.
Yes. Thank you, Leong. Let me share. Okay, so I think you all can see my screen. So good afternoon, first of all. And Leong and me knew each other through badminton in Cambridge. So he was working with in Benki's lab on ribosome and crystallography. I was doing mostly EM and membrane proteins. So we met through badminton. He taught me a lot of how to play a good badminton. Oh, okay, so today I thought I will speak about how electron microscopy has transformed from the old age of just looking at things at low resolution to very high resolution. I am affiliated with NCBS in Bangalore, and there are 3 institutes in the campus. One is NCBS, then we have INSTEM, which is Institute for Stem Cell Science and Regenerative Medicine, and then another institute called CCAM, which is housing a lot of incubators to promote startups and make translational research from basic research to clinical side. So these 3 institutes are together called Bangalore Life Science Cluster. So there's a good community here doing a lot of different basic science, and that has promoted a lot of collaborations also. So most of the slides, this was made by myself and Richard to give more on the basics of how cryo-EM works. And I start with asking why we want to use electrons. So here is a spectrum showing different wavelengths and what we can see. So when we use a light microscope, you see just objects with low resolution, but the electrons, for example, a 300 kV electron is very small and has a wavelength of about 0.02 angstrom, which means that in principle, one should be able to see atoms. This can be realized in material science, but not in life sciences. So just to give a basic background, so we have electrons in electron microscope. I don't need to say that, but if you look at the wavelength in light microscope, it's between 300 to 600 nanometer. Even with the 100 kV electrons, the wavelength is very short. Instead of glass lenses, we use magnetic lenses in the EM, and the disadvantage is that we have to use vacuum to look at specimen, and we are not able to observe live cells. So those are kind of advantages and disadvantages of EM. And towards the end of the talk, I will explain how we can combine both this light and EM to get the best of both. So a typical electron microscope looks like this. So this is how a old Philips microscope looked. Philips used to make the initial microscopes. Then they decided that they will focus more on medical instruments like tomography, like PET scan and MRIs and light bulbs. So Philips then became FEI. Is based in Eindhoven in Netherlands. So when you remove the cover, you see something like this. You have a filament from where the electrons are coming in, And there are a series of lenses which kind of focus, and then when it interacts with the specimen, it magnifies, and you see either on the screen or on the camera. So this ray diagram is very similar to what one usually sees in light microscopy also. After interaction with the specimen, there's a series of lenses which then make the image. So in the old days, when we had such microscope, it was in a room which was dark, because you had to see the sample here in the screen. And because it was not covered, which meant that you can't just open the door and get inside to prevent vibration. So what the companies have done now is to put an enclosure. So this is now the most what we call an advanced system, where the same thing what we see here is inside this box. And this prevents, for example, when you open the door, you can just go in if the data is being collected. So this enclosure is also proof from sound and minimizes some of the or increases stability. So this has made a big difference in terms of automation. So electrons interact with a lot of things in an atom. So because they are negatively charged, they interact via Coulomb forces. They interact with the valence shell electrons to create low angle scattering. They interact with the inner shell electron to create medium angle scattering, and also with the nuclei, which gives high angle scattering. So because they interact with everything in an atom, they scatter quite a lot when compared to X-rays, which interact only with electrons. And if you take neutrons, they interact only with nuclei. So because electrons interact with everything, the map we get is called Coulomb potential map. In contrast, in X-rays, you get electron density map. So because they interact very strongly, this means that you also have a limitation on how much thickness you can have a look. For example, if you have a very thick specimen, let's say a bacteria, which is about a micron in diameter, then most of the electrons will be absorbed and you won't be able to see much of it. So thus, in material sciences, generally people use about 500 angstrom thick specimen. In particular, if you have a heavy element like, for example, a steel or a semiconductor or so on, so they are limited to this kind of thickness. In biological sciences, we have mostly light elements like carbon, nitrogen, oxygen, and so on. So the thickness can be increased slightly, and electrons can still pass through. So when electrons interact with the specimen, we get a lot of signals. And much of the beam which comes and interacts with the specimen is direct beam, about 80% of it. And then there are other things which we will not discuss. But the most important are the elastically scattered electrons, that's about 5%. And you have inelastic scattered electrons, this 15%. So these electrons, which elastically scatter, maintains energy. There's a change in phases, and these are the ones which give signal. And the 15% of these electrons result in loss of energy, meaning they cause radiation damage. So one hopes that we tend to avoid this and try to increase this. So when electrons interact with specimen, electrons are absorbed. This is how we get one of the contrasts, which is called amplitude contrast. And there is scattering over small angles, which gives rise to phase contrast. This is shown here in this picture. So you have the direct beam, which comes through the lenses without interacting with the specimen. And when there is a small absorbance of electrons, there is a difference in amplitude, and also a small scattering, which gives rise to change in phases when compared to the direct beam. So in life sciences, these both are seen, but the phase contrast is the one which dominates in biological cryo-EM. So the amplitude contrast is between 4 to 8% or 7%, depending on the setup in the microscope, but the phase contrast dominates. So electrons, because of the wavelength, they are very good. One should be able to get very high resolution, but there are few issues which one encounters. The biggest issue is radiation damage to the sample. This is because they are very energetic, and to some extent we can reduce the temperature of the sample to liquid nitrogen temperature or liquid helium, so that the onset of radiation damage is slower. Using this temperature doesn't remove the radiation damage, but it's slower. Then we have something called specimen charging. So we look at life science or biological specimens on ice, because water is the most major constituent. And when the beam hits or the electrons hit the specimen as well as the surrounding material, they tend to release some charge, and this tend to blur the image. And these are very expensive machines. Some of them cost to about $4 to $5 million dollars. And to maintain that and to keep feeding with liquid nitrogen and also to run it, it's a bit expensive. So these are some of the issues with electrons. And when I mean by radiation damage, so here in this movie, you will see that this is a frozen sample of just water. And as the electrons is being bombarded on the specimen, you will see that this ice, which was there, eventually pops out. So this is what happens to your protein samples or macromolecules when you put on the grid and visualize by electron microscopy, that they get damaged. So which means that you will have to average a lot to get enough information beyond the noise. So in life sciences, there are multiple things one can do with electron microscopy. You can look at molecules, a single molecule, which we call single particle cryo-EM. So here is an example of hepatitis B viral core particle. You can see individual viral particles here. This, after averaging, you can get map like this. Or you can look at things which are helical arrays or helical tubes, such as in this example, the acetylcholine receptor. Or you can have actin or microtubule, which also form helices, and you can do helical reconstruction to get a structure like this. Or one can look at two-dimensional crystals. Here is an example of bacteriorhodopsin, abundant protein in Halobacterium, and which Richard Henderson used to work during 1970s and 80s. And using diffraction and electron crystallography, you can get then a structure. Or you can also use tomography, where instead of doing all these things where you isolate the protein, you can look at, in this example, bacterial cell, and also look at things like ribosomes and cytoskeletons of the bacteria by tomography and then averaging it. So this just shows that you can do a lot of things with one instrument. And currently, what are the possibilities? So if you have a single particle or single molecule, I just put here about 55 kilodalton to about 40 megadalton, and one can reach a resolution of about 2 to 3 easily, or sometimes less than 2 also. This number here, I keep changing because as technique evolve, this number goes down. So currently I put less than 40, we cannot orient. So small molecules like lysozyme or albumin, which is about 40 kilodalton, you can see on the images by electron microscopy, but the orientation or determining the structure is still a challenge. But this number is likely to go down. In terms of 2D crystallography and helical reconstruction, there is no limit on the size, and you can also reach very high resolution. But getting good crystals or ordered helices is a difficult thing. So most people now have abandoned this, and but they do. So when your specimen is helically ordered, then this is now becoming very powerful. But 2D crystallography is not studied much at the moment. Then you can also have tomography, where instead of taking single image, you do a tilt series. And these raw tomograms are noisy because of the low dose we use, because you don't want to damage the specimen. And these raw tomograms are very low resolution, but within these tomograms, you can have objects like ribosomes or proteasomes, which then you can do averaging and get very high resolution. I think the highest now has been about 3.4. This is still not high throughput, and all the other things are high throughput, meaning you can put a specimen, automate it, and just leave it for a long time. And the last thing is called microelectron diffraction, where small 3D crystals which you are not able to see, but you can put in the microscope and do the same as you do in X-ray crystallography, like collect diffraction data and then get a structure. So this is now also a popular technique where nanocrystals of peptide proteins can also be obtained at high resolution. So this slide just to show 3 different membrane protein complexes of different size. You can see here the years 2012, 2013. They were just blobs, meaning they just had a overall envelope, but there was no structural features. But within short period, very high resolution information started to come, as you can see here in the helices, and also the ion supercluster in this electron transport, the enzyme in the electron transport chain. So in my talk, I will be explaining how this transition from blobs to high resolution was possible. The other advantage of electron microscopy is to study heterogeneity. So this is an example of ATP synthase, which is a nanomachine. So this is just a movie of one sample which was frozen in EM, where you have multiple population. And so such heterogeneity can be classified computationally to separate them out. So in this example, we had an inhibitor bound to the F1 region of the ATP synthase, and I've color coded the central stalk in blue, green, and magenta, and the inhibitor in cyan. You will see that the computer is now able to classify the whole population into three different states, because it's a rotary motor. You have a rough rotation of about 120 degrees to go from one state to the other. This can be seen in this model here. The inhibitor is bound at different interface and locking. So this is an important thing because most of the biological macromolecules are dynamic because it's needed for their function. And by combining both electron microscopy and computational methods, you can try to get multiple structures from a given sample. Okay, so this is an introduction. So how did we get there? So in the next about 5 to 10 slides, I will tell how cryo-EM has developed. And much of this was based on the review myself and Richard wrote in 2016, when I was still at the MRC in Cambridge. So the first developments happened both in academic as well as industry. So one of the important thing is the development of plunge tracing. This is how we make the sample. So in hindsight, it's easy to think that you are just taking a sample and just plunge tracing, but it was a lot of work in 1980s to get this perfected. And in terms of microscope development, we had very good source. So instead of having a tungsten filament, now we have a field emission gun in the as an electron source, which gives very coherent beam. And we have stable cold stages and better vacuum. So vacuum is important because when we put samples which are frozen, it's necessary to keep the vacuum very good so that you can collect for longer time. The same is true for how we visualize the sample or detect. So starting from film, now we have direct electron detectors. This has made a big difference. And in computationally, we have very sophisticated algorithm that use both GPUs for faster computation as well as maximum likelihood methods like rely on. So there has been a lot of advances. And I will start with these images. So these were taken by Helmut Ruska, his brother of Ernst Ruska, who made the first transmission electron microscope. So Helmut Ruska is a medical doctor. He took samples and air dried and put it on a EM grid. So this is a bacteria, and you see there is a bacteriophage sitting on the bacteria. So these were some of the first images taken of a biological specimen on an electron microscope. And subsequently, similar viruses like T3 and T7 were taken by others. You can see the viruses here. So these were all air dried specimen, meaning you just take the sample, dry it, and then have a look. The next technical advancement came by the introduction of a process called negative staining. So this was introduced by Sidney Brenner at LMB in Cambridge. So what we do here is you have a protein sample, which we, for example, these are all viruses. We applied to it on a EM grid. Then we use a heavy metal like uranyl acetate or tungstate, which will form an envelope around the protein. So that's giving contrast. So here we are seeing the envelope of the negative stain or the grain and giving very nice contrast, and one could see the arrangement of the viruses. So these are textbook images you can find in many of the textbooks. And so this was very nice because we could see a lot of the structures. And during this time, also at the LMB in Cambridge, Aaron Klug and David D. Rosier developed the computational methodology for obtaining a 3D structure from a 2D projection. In 1982, Aaron Klug was awarded the Nobel Prize for developing this technique. So here is an example of a bacteriophage again. And what they did was to formulate a mathematical and computational method to take this tail from the phage to get a structure, a 3D structure like this. So this was a helical specimen, and subsequently using the same or similar principle, Tony Crowther was able to also get a reconstruction of spherical viruses like shown here. This is tobacco resistant virus to get a structure like this. The same principle or image processing to go from here to here. So one thing to realize is that because the stain which is coating the samples here, this limits the resolution, and people realize that with negative staining, one cannot reach beyond about 20 angstrom. So during this time, Bob Glaser, who is based in University of California in Berkeley, he was trying to understand from theoretical perspective what will happen in terms of current density or electron dose to amino acids or nucleotide and how they fade. For example, nucleotides, because they have phosphate, they fade a bit slow, but amino acids like valine, they fade faster. So what he meant, or he concluded, was that it is possible to get higher resolution if you average enough, but also use low amount of electron to avoid the radiation damage. And Bob Glaser and Ken Taylor were the first people to go away from negative stain and do cryo electron microscopy. So what they used as a specimen was a thin 3D crystal of catalase. And so this is a diffraction pattern where you can see that the spots go farther away. But this was done during the time when there was no the holders or good vacuum. So they had very limited time. So they were able to at least show that in principle, if you do a good cryo preparation, you are able to preserve the structures in crystals. So during this time, also in 1980s, Jacques Dupassé was recruited to EMBL Heidelberg by John Kendrew to develop the process of what we call cryo electron microscopy. So this is one of the first instrument which Jacques Dupassé and his colleagues used. So you just have a forceps and grid here. There's just a rubber band, and there's a ethane here, which is used for freezing. And this is a bit more advanced version where you can see the same thing again, a EM grid, a forceps, which then plunges here. So what Jacques Dubussier and his group did was to study what happens to water. And this is a very nice review which they have written, and it's very useful to read. What this image shows is 3 types of ice. You have hexagonal ice, you can see from the spots here, and when this is slightly heated, then you get cubic ice. And this is an image of amorphous ice or vitreous ice with polystyrene spheres here. So when we do electron microscopy, cryomicroscopy, the idea is that you tend to avoid these structures of ice, but towards go to amorphous ice, so that you are able to see your molecules better. So this is an instrument which we used to use in Cambridge. It's a very simple instrument. You can see there is a rod with the forceps, and here we have liquid nitrogen, and this inner chamber has liquid ethane. There is a EM grid here. So once we put the sample and take away the excess fluid, we just plunge it in ethane. Ethane is used because it gives faster rate of freezing. And then this is how a typical image, low mag image looks. So you have holes here, and the hope is that we have a thin film of ice with the particle, which is shown here. So you have particles, and then these are the edges, and then we take images here. So using this technique in 1984, Jacques Dubussier and his colleagues, they took first images of this virus. This is the adenovirus. So this was the first time somebody had taken a image of a virus, and this was in the front cover of Nature. So this was in 1984, but it took them about 7 years to get a 35 angstrom resolution map, because that time the computational methods were not developed. They had to develop. And the same virus, which was done in 2010 with better microscopes, but in the same kind of medium, which is film, you could already see here how the features of the virus when compared to this is very visible. And using a lot of images, Hong Zhou and his group at UCLA were able to take this and get a resolution of about 3.6 angstrom. So this was on a photographic film, and you can see clearly the separation of the beta strands and also the side chains. So this was a big improvement, and mainly because of the microscope stability and the coherence of the peg and so on. During this time, Richard Henderson, he wrote a review. It's very illuminating on comparing neutrons, electrons, and X-rays. In this review, from first principle, he postulated that one should be able to get high resolution images of different sizes of macromolecules with as little as about 12,600 images or molecules to about 3 angstrom. This is what he kind of predicted, and he gave explanation of how we can achieve. So he kind of pointed out 3 factors which one needs to overcome to realize that. So one of them is radiation damage. As I explained, this is going to be there. And then we have beam induced movement and charging, and then signal to noise, which is how you detect the specimen. So a lot of improvement in the recent times has been mostly on these detectors. Also, we have had improvement in how we make specimen, but I'll just give a couple of slides on how we used to detect. So in the old days, when I was still a student, we used to use what we call a photographic film. This was on a camera chamber where electrons hit this the film, and then you can go to a dark room, develop it so that you get a image of whatever you are looking. So this was kind of superseded by CCD cameras or charge coupled devices, where the incident electron is converted into photons and then back to electrons. So these two were in the old days very good, but the problem was that they were very slow and they had very DQE, low DQE or detective quantum efficiency. This means that the amount of signal that's coming in and that comes out is low. So in particular, in the case of photographic film, we had the issue that you had to go to the darkroom to develop. So until you develop the film, you will not know if the image was good or not. So you take about 50 images in a given session. You have to develop before you know if it was good. So this was a limitation. So in recent times, there has been development in these detectors. Now we have something called direct electron detectors, which are based on CMOS technology, complementary metal oxide detector sensors. So here we have a slightly p-doped silicon wafer, and this is a sensitive layer. So when an electron, so this is a simulation done by Greg McMillan at Cambridge. So when you have a 300 kV electrons coming in, it creates electron hole pairs, which are then detected by this N-type diodes, and this substrate is here for just support. So this backscattering is just to show how one of the events, how energetic they can be. So the bigger advantage of this detector is that they have a higher signal to noise ratio as measured by the DQE, and they also operate in a rolling shutter mode, which allows recording of movies. So this is just an example of a curve which shows DQE and then different detectors and how they are. You will see that at lower spatial frequency, they are very high, very good. And then as it goes to the higher spatial frequency, they kind of fade out. So this slide shows how we have improved for a given specimen. So this is a beta galactosidase, which is used as a test specimen. We started at 25 angstrom in 2005. It got better to 11, and in 2 years it went to about 6, and then within a year with development of detectors, we went to about 3.8, and this resolution is in 2015. Now we have better than that, where you can clearly see the side chains. So the basic image processing principle in single particle EM is shown here in this nice cartoon, where you have different views of this particular object, the house. The idea is that you pick these particles and then you classify to get same views averaged out, and then we try to estimate a 3-dimensional structure. So to get this structure, we need to determine 5 parameters of each of these particles, 3 Euler angles and XY translation. The higher the accuracy, the better the structure will be. And this is a typical workflow where we start from images or micrograph. Then this is something I skipped. We have to estimate what we call contrast transfer function. Because to get contrast of this biological specimen, we modulate the objective lens current, which gives these tone rings. This is an important parameter we have to determine accurately to get very high resolution. After we estimate, we pick particles, you average to see how heterogeneous, homogeneous they are. Then we refine, and in case of dynamic molecules, you classify in 3D to get, for example, in this case, ribosome, to get different states or different composition, and you interpret. The whole workflow here is, the aim is to average so that we increase the signal to noise and interpret the map better. So this is one of the molecules which I used to work, where we start with micrograph. We do two-dimensional class averages to see how they are, and then using this particle to get these kind of maps where we determine the orientation, refinement, and reconstruct. So in reality, the cryo images have a lot of noise because we use very limited dose. So they are very noisy. This is like looking at the evening sky and trying to see how many stars are, where the stars are, how far they are, what is the composition, and so on. Something similar, we have a lot of noise. So as I said, we need to defocus or change the objective lens current to get contrast. So this introduces what we call artifacts, the contrast transfer function. If you had a good faceplate, then this is not needed. We will get away. The other thing we don't know is how the particles fall on the grid. So we have unknown orientation and classes. So this, in mathematical terms, we call inverse, incomplete, and in-post problem, which many of the algorithms try to tackle or try to solve. So this is an example of what we get when we typically do a cryo-EM. So this is catalase, which is about 230 kilodalton. This is well studied. So it's a good specimen for cryo-EM because it tends to behave very different. So you can see here, there is no ice and the particles tend to kind of cluster together. They don't like it. And you can get pseudo-crystalline arrays. So if one tries to process this kind of samples, then you get what we call anisotropic map. You can just see the overall shape of the molecule, but it doesn't give meaningful interpretation of the map. So then you work harder to get more uniform and different views, like shown here. So then you get very nice classes, and from there you can get a very nice map and also look at the ligands. So this is a common problem that one encounters, and then one tries to work through like in any other technique to get better map, better specimen. So when I talk about movies, when we look at one of this image, this is a summed image. This is of hepatitis B viral core. This is about 4 megadalton. So a movie consists of different frames. So these are single frames with defined dose. And when we sum them, we get this summed image. So because we have these different single frames, now it is possible for us to look what has happened during the exposure. For example, this is a single frame with 0.6 electron per pixel. And this is an average of about 9 frames where you start seeing particles. So because you have this information, you can now computationally track each of these particles to see what has happened during the exposure. So here all these particles are marked, and then you can follow the trajectory to see what has happened during exposure, and this can be computationally corrected. So when we did this the first time, we found that you can plot either a B factor or a resolution. In x-axis is number of frames or number of electrons. What we found was that there was an initial fast movement which made the images very poor, and then we also had radiation damage, which kind of which we knew which was happening. And using this information, then we could actually increase the resolution from about 5 angstrom to about 4 angstrom just with 1000 particles for this hepatitis B viral core. So these are the computational processing, not with the specimen. And this is what you get this is about 4 angstrom map with 2 hours of data collection and less than 1000 particles. Because it has symmetry, it's very fast to do this. And this movie just shows the whole 3D map where you can start seeing the side chains and also the helices in this virus. So it's 1:25. I got about five minutes. So after I came to NCBS, we set up this facility. We have got multiple structures. In the last 5 minutes, I think I will take one example from our facility to go through how we use cryo-EM to understand biology. So this was an enzyme which we found that it has a novel iron-containing active site in the enzyme. So this is an enzyme called dimethylformamidase. This cleaves dimethylformamide. Is an industrial solvent used in leather industry in particular. And this is a very stable compound. But some of the microbes, as we know always, that they come up with an enzyme to degrade this, and they can use DMF as a sole carbon source. So this was identified as an enzyme with 2 polypeptides. There's a large and a small subunit, 85 and 15 kilodalton. And it was known that in 1986, they are likely to form oligomer and highly stable enzyme. So when we started, we wanted to know how these polypeptides are arranged, what is the active site mechanism, and substrate specificity. So DMF, when degraded by the bacteria, can be taken either as formate and dimethylamine and then assimilated into the normal metabolic pathways. So when I first looked at this enzyme in the EM, I noticed that there were 2 populations marked in blue and red here. You will see that there are 2 populations. And you can, so because you can visually see, computers also can classify. You can see here now some of them are marked here. You have the bigger molecule and the smaller one. And this can be separated. So you get two structures. In this case, they are of the same molecule, but if in case you also have two molecules, then you can separate them. So eventually we found out that this dimer and tetramer equilibrium of the same enzyme depends on the salt. So the high salt, it tends to form tetramer, and the low salt, it forms to form more dimer. And we got map. So the dimer is alpha 2 beta 2. It has four polypeptides of a large and small subunit giving to 200 kilodalton. So you can see the alpha and the beta subunit. And the tetramer is made of two of these dimers. And these are some of the density, Coulomb potential density. From the EM map, you can see nicely the side chains and the beta strands separated. And when we model, we found that this enzyme has some novel folds. For example, this beta subunit has a novel fold, and also where the ion, the active site is also very novel. So after we modeled the polypeptide, we found that there was an extra density here between these two tyrosines and glutamate. We thought it could be a metal ion, and we modeled ion atom here. And this water is just to finish complete the coordination of the ion. The validity of the ion was confirmed by X-ray spectroscopy as well as by EPR. So now we have got a map to about 2.2 angstrom for the same enzyme. Now we are able to confidently build water molecules. In particular, this is important in the active site because enzyme mechanism can be explained by using only through waters or mostly through waters. You can see here the active site with a lot of water molecules. Now we are able to build about 100 to 120 water molecules very confidently. And we have been also making a lot of mutations around the active site. And this mutation here, 693, which is phenylalanine, which is far away from the active site, we find that mutating to alanine, it has the ion bound but has no activity. But when you mutate to tyrosine, it increases in activity. So currently what we are trying to understand is why is this phenylalanine required, and what is the role of all these residues here, because most of them lose the ion atom in the active site. And another interesting thing we found was that in the active site, we also tend to see extra density. So this is in a mutant structure where we see a long density. At the moment, we are not able to interpret because it could be anything from the buffer or anything from where the enzyme came or growth. So this will need possibly higher resolution or maybe complementary technique to understand what is the density here. So we are also trying to see how we can remove this density by different methods. And because this enzyme is active only at higher temperature, we can freeze at low temperatures and try to trap substrate by mixing, also using inactive mutant to see if we can get a substrate bound here. And this movie shows how the molecule is here. Here is an pore or portal where we think the substrate enters, and these are the two. This portal is at the interface between the small and the large subunit, and we think that the substrate goes in here and the active site is in underneath, and this is where the substrate goes and the active site is buried here. So when you look at the active site, you'll see that there's a lot of hydrophobic groups here, which probably explains how only small substrates like DMF is accepted, and larger substrate, when you have a big group like a phenyl or a benzyl ring, is not a very good substrate. So in summary, this particular enzyme, we built de novo the map, the model from the map from the EM map, because there was no structure from the PDB. So we built everything from scratch, and it has an ion which is coordinated unusually by tyrosines and glutamate, because ion generally is coordinated by cysteines and histidine. And we think this is how the substrate enters here. Leong, can I take 5 minutes and finish?
Yes, please.
Yeah.
Yes.
Okay. So that was one example of what we have done in Bangalore. So I'll finish by giving some thoughts on where the cryo field is going. So what we have achieved is very nice, because there's a lot of structures which was not possible before, now we can do. And I go back to this review article which Richard wrote in 1995, which he calculated you needed about 12,000 asymmetric unit to get about 3 angstrom. This number has been revised. Now we think that you need, this is by theory, not practical, this is about 800 particles. So this number is based on what we call B total. B is for B factor, and this is dependent on how good your image and how good your image processing or orientation determination, which then gives rise to these numbers here. So the lower the B factor, the higher the resolution, and the lower the numbers would be. These are shown for different molecules in these plots here. You will see that this is experimentally determined B factors and the number of particles, and here you have the theoretical estimate. So what this plot gives or tell us is that if you reduce the B factor, you will get high resolution without averaging a lot of particles. Lot meaning millions of particles. So that's what these plots show. So why do you need so many particles? So these are shown in, These are shown here in 4 of these graphs of different samples. You see that here in the first electrons, there is a lot of lower or a lot of higher B factor, or in this case, you can see there's a lower resolution, and then it plateaus and then it kind of goes down. This is radiation damage. So we think this is one of the reasons why we need still more particles. So to realize the full potential of electron microscopy, so currently we have these kind of detectors. We hope that in the future we will have a detector which is also very good, has a higher DQE at also at higher spatial frequency. And this B factor, if you can solve this initial movement and get this B factor like a straight line instead of having this lower B factor and then plateau, and so then I think we will have achieved the full potential of cryo-EM, where you can get multiple structures in a given day very fast. And Chris Russo at Laboratory of Molecular Biology in Cambridge has been developing a lot of grids, better grids using gold and smaller holes, where they are trying to reach this particular line here. And I think in the recent paper, they have shown that it is possible now to get better images using smaller holes. And what I didn't go in detail was tomography, which I think has a lot of features apart from single particle. So in tomography, instead of taking one image, we take a tilt series. And basically you have a tomogram, and once we have, we can locate the objects and we extract them. And similar to what we did with single particle, you align to reference and then average, and then you get a reference. You go through iteration to get higher resolution. So this now is becoming a more popular and technique where you can study things within the cell, and combined with correlative light EM. This is from a review from Grant Jensen's group, where you can label a protein, look it in cryo light microscopy. Here is a hole, and you can see the fluorophore. Then you take it to the EM, you take a tomogram, then you can do subtomogram averaging and get a pseudoatomic structure. So cryotomography and combining both light and electron microscopy, I think has a lot of bright future. And these are some of the examples of things where sub-nanometer resolution has been reached or becoming possible from tomography, not by single particle, just by taking things from the cell. And I end by giving this last slide where what are the potential of electron cryomicroscopy? Large labile complexes now are possible, in particular a lot of eukaryotic macromolecular complexes and membrane proteins. So the bigger advantage is that you can also look at multiple structural states, not one. And things like filaments, actin microtubules, and amyloid fibrils are now possible to get very high resolution. And within the cell, ribosome, proteasome, viruses, and membrane proteins are now being looked into, and also thin 3D crystals by electron diffraction. And the recent papers in 2020, 2 of the papers now show that you can get to as high as about 1.2 angstrom from single particle EM. So this is of a tested specimen like apopharatin, but getting this kind of high resolution gives a lot of confidence that this method has a lot of potential. Okay, I end by thanking the funding agency, which is the Department of Biotechnology of India, which funded the BeLife grant, which gave us this microscope. This is the web page where we have information on the facility and how we collect data and so on, and all the funding from NCBS and INSTEM. And I'll be happy to take any questions you have.
Thank you, Vinoth, for the very good overview on the development of cryo-EM from the very beginning, from how scientists done in the air dry, then negative stain, until today we have atomic resolution up to 1.2 angstrom. I think that shows how powerful the electron microscopy with the cryogenic, incorporating the cryogenic to look at the biological sample that we all would like to see how things or how life work, and also very nice, beautiful structure of the methyl formylase enzymes, and seeing the binding of the ion in the active site likely. So I think that gives us open our eye, and as a whole, to see the potential of cryo-EM in solving or understanding the biological events that we all are interested in.